photosynthesis or in a dark phase of photosynthesis. The Calvin cycle, also known as C3-way pathway, is a crucial part of photosynthesis. This process by which plants convert energy into chemical energy in the form of sugars. This cycle is named after Melvin Calvin, who along with his colleagues used radioactive carbon isotopes to trace the path of carbon during photosynthesis. The Calvin cycle is a complex series of reactions that occur in the stroma of chloroplasts, the green organelles found in the plant cells. First things first, we look at the methods of studying the Calvin cycle. There are three types of methods to study. First is radioactive carbon method. It is crucial method for this. It is involved using radioactive carbon isotopes which behave similarly to their stable counterparts. These isotopes act as markers, allowing the researchers to track the movement of carbon through the various compounds involved in photosynthesis. Second is chromatography on paper. This technique separates different compounds based on their properties, allowing researchers to identify and quantify the various molecules involved in the Calvin cycle. And the third method is radioautography. This method uses the radioactive decay of isotopes to create image of photographic film, revealing the location and distribution of radioactive compounds within the plant. We look at the Calvin cycle as step-by-step breakdown. Calvin cycle includes the three steps in it. First step is carbon fixation, or we just call it carboxylation. The first phase involves the fixation of carbon dioxide, that is sucks from the air. Carbon dioxide by the enzyme ribulose biophosphate, carboxylase or oxidase or Rubisco enzyme. This reaction forms an unstable 6-carbon intermediate which quickly breaks down into two molecules of 3-phosphoglycerate or 3-PGA. Second step is reduction. After the first step, second is reduction. The second phase involves the reduction of 3-PGA to G3P or 3-phosphate using ATP and ADPH products of the light-independent reactions. This process requires energy and reducing power to convert 3-PGA into a more energy-rich compound. And the third last step is regeneration. In this step, Calvin cycle regenerates the RuBP or ribulose biphosphate. The third phase involves the regeneration of that thing, the acceptor molecule for carbon dioxide. This process uses 5 molecules of G3P and requires ATP to complete the cycle. As the first we said, Rubisco, it is the enzyme that is plays a very crucial role in Calvin cycle, especially in the first phase, in the carbon fixation phase. Rubisco, the enzyme responsible for carbon fixation, it is a key player in the Calvin cycle. It is the most abundant protein on Earth and is found in large quantities in chloroplasts. Rate of the photosynthesis. Here are the table that is faces us to the feature and the description of the things. Feature first is function. Function of Rubisco, the enzyme, it is catalyzes the fixation of carbon dioxide. It is in the stroma of the chloroplasts. Abundance is the most abundant protein on Earth and the rate limited step, slowest step in the Calvin cycle. The fate of G3P. The G3P molecules produced in the Calvin cycle have two possible fates. One molecule of G3P exits the cycle and used to synthesize sugars, amino acids, and other organic molecules. The remaining 5 molecules of G3P are used to regenerate RuBP and ensuring the continuation of the cycle. First is G3P exit. One molecule of G3P exits the cycle and used to synthesize sugar, amino acids, and other organic molecules, and it also regenerates RuBP as it faces it. Also, we have the C4 pathway. While Calvin cycle is primary pathway for carbon fixation in most plants, some plants, particularly those adapted to hot and dry climates, use a different pathway called the C4 pathway. This pathway involves an initial fixation of carbon dioxide by a different enzyme, resulting in a 4-carbon compound. The C4 pathway is more efficient at fixing carbon dioxide in hot and dry conditions. We faces the difference between Calvin cycle or C3 pathway and C4 pathway. C3 or Calvin cycle is the initial fixation of carbon dioxide by Rubisco, forming a 3-carbon compound or 3-PGA, by a different enzyme forming a 4-carbon compound. The importance of Calvin cycle. Calvin cycle plays a crucial role in the for the life on the Earth, and also it is the primary pathway for converting light energy into chemical energy, providing the foundation for the food chain. The sugars produced by Calvin cycle are used by plants for growth, reproduction, and other vital processes. These sugars are also consumed by animals, providing them with the energy they need to survive. Here are the main types of things that Calvin cycle is more important. First is plant growth, as well as the sugars produced by the Calvin cycle are used by plants for growth, reproduction, and other vital processes. Second is food chain. These sugars also consumed by animals, providing them with the energy they need to survive. And the third is life on the Earth. The Calvin cycle is essential for the life on Earth, providing the foundation for the food chain. The Calvin cycle. There we talk about ATP and ADPH that are very crucial for the Calvin cycle. It requires a significant amount of energy to function. To restore 6 molecules of carbon dioxide to the level of carbohydrates or a glucose, 18 ATP molecules and 12 ADPH are needed, or overall there are 30 molecules of the energy. This translates to 3 ATP molecules and 2 ADPH molecules per carbon dioxide molecule fixed. The energy for this reaction is supplied by the light-independent reaction of photosynthesis. ATP, there are 18 ATP molecules are required to restore 6 molecules of carbon dioxide to the level of carbohydrates or the glucose. NADPH, 12 NADPH molecules are required to restore 6 molecules of carbon dioxide to the level of carbohydrates or a glucose. The regulation of photosynthesis. Here we talk about the Calvin cycle and its regulation of photosynthesis in the dark stage of it. The Calvin cycle is cyclically regulated to ensure that it operates efficiently and produces the appropriate amount of sugar. This regulation is influenced by factors such as light intensity, carbon dioxide concentration, and the availability of ATP and ADPH. The enzymes involved in the Calvin cycle are also subject to regulation, ensuring that the cycle is responsive to changes in environmental conditions. Understanding the regulation of Calvin cycle is crucial for optimizing plant growth and productivity. By manipulating environmental factors and understanding the mechanisms of regulating, scientists can improve crop yields and enhance the efficiency of photosynthesis. After the lecture, we watch a small video about the Calvin cycle. So you have to more think the process of it.
The light reactions in the thylakoid, which are summarized by the following equations, require sunlight and water among the reactants and generate oxygen, ATP, and NADPH as products. ATP and NADPH are then used in the Calvin cycle, which occurs in the stroma. This is where all the synthesis occurs. Unlike the citric acid cycle, which is catabolic, the Calvin cycle is anabolic, building organic molecules from smaller components, requiring energy to do so. This happens in 3 phases. Phase 1 is carbon fixation, which is catalyzed by an enzyme called rubisco. Rubisco captures CO2 from the atmosphere and attaches it to a 5-carbon sugar called ribulose bisphosphate. The resulting 6-carbon molecule is unstable and splits into 2 molecules of 3-phosphoglycerate. Phase 2 is reduction. Each of these molecules receives a phosphate from ATP and is then reduced by NADPH, after which it loses a phosphate to become glyceraldehyde 3-phosphate. Some of this G3P is the output of the Calvin cycle, going on to other pathways that generate glucose and other organic compounds. And some goes back to generate more RUBP and start the cycle over again. To generate a net output of 1 G3P, the cycle needs 9 ATPs and 6 NADPHs.
Thanks for your attention, and hope you have the best to understanding the Calvin cycle.